So let's take a look at some of these. So if you look at the tabs here, and like I said, if you learn how to use IPA, you're going to be a big deal in bioinformatics. You've got your summary page, just kind of your greatest hits of all your different categories, right? You know, upstream regulators, blah, blah, blah. You also have canonical pathways. Click on that second tab. You'll notice too, though, is that a lot of these, when we start looking at the other genes, like this FAC signaling comes up. Very significant. The problem is we're only getting 2 molecules out of this, or 2 genes in our gene list. Remember, though, we only have 25. And I'm going to tell you right now, that's one of the limitations of having a very small gene list, is that your group that you're testing to see if you can pull out is extremely small. You know, the draws that you get, so it's very difficult to do the statistics to see if anything's overrepresented. But you can open up any of these, click on this bar. Well, actually, click on it. So here are the 2 genes in our gene list associated with this pathway. Let me explain this chart here. Given here is the negative log of the p-value of its significance. Again, this is based on a Fisher's exact test on exactly the slide I showed you, right? What are the odds, the probability that we get all these genes associated with this particular pathway by random chance? You'll notice that this threshold here, this is the line where it's considered significant. So at about 1.33, I think, is equal to a negative log of 0.05. Does everybody understand that? So if the bar is higher than this line, then that means it's significant. Everybody got that? This down here is the ratio. This is telling me, based on this, that in this FAC signaling, there are 87 members. You can see if you put it on that line, there's 87 members in this pathway, and we got 2 of them. So we're basically pulling out 2% of that. Pretty close. Anything else on this? You can click on the bar. So we clicked on the bar. You can open the pathway. Anything that's colored was in your gene list. So here we got this graph and this calpain. I actually looked these up. These are totally related. Like, this is a decent data set. As I'm going through and I looked at their paper, I'm just like, why didn't they talk about any of this stuff? Like, there's a lot of good stuff in this. Again, it's like being the accountant for all of this like data, and you just yell at the CEOs like, why are you doing that? Don't launch that product. Launch this one. All right, you can close out, but you can kind of see that. Okay. And we can manipulate those. I'm going to show you more when we get to the ANOVA stuff, the genes that were affected by date and cell type. You can go to disease and functions, and we're going to see this here. This is basically showing you all the genes or the biological functions that were overrepresented in your list. We also have the upstream regulators. And this is kind of a cool page. This is where you find those rocks. And we can actually sort this by target molecule. I'm sorry, I'm kind of like going fast. Did I lose anybody? Everybody on the same? Okay, cool. So we can see some of these things have 2 targets in our 25 genes. Which, I don't know. Like I said, this is a small list. Here's where things get interesting. Go to networks. Okay. You'll notice this network here. Everybody got this? It's got a score of 45, right? Everybody see that on yours? You don't have a network with a 45? 25? Huh, interesting. But that was the 25 genes, I thought. Okay, I'll check that in a minute. Here's what next works are. If you click on this, click on your top 25. What this does is it groups the genes not based on a pathway, not based on a biological function, not based on its connection to an upstream regulator, but its connections with each other. You can see these lines here. If there is a solid line, that means that there is a direct biological relationship. And I can double click on any of those lines and view the paper or the manuscript where that comes from. Calpain has a member, Calpain 11. And I can go there and click on this and it'll bring me to the paper. Maybe. There it is. Oh, this is just the thing. Never mind. Would be a better one. We'll do it with the other ones. This is a very good page because I would say, and I'm going to tell you right now, that most of the pathways out there are BS. Okay? That some of them are like 20 years old. And that, you know, these were a bunch of experts looking at different organisms and different tissues that kind of got together and said, let's group them all in this pathway. Right? This is kind of like, you know, if you've seen my lectures on the, you know, kind of looking at gangs, you know, and these kind of network stuff, you know, this would be, I don't know, where was I going with this? I just taught another bioengineering class right before this, so I'm kind of confused on what I tell them and what am I telling you guys. What this will do is basically what you can do is that the pathways are not very accurate. That if you really want to get the true signaling in your network, you kind of have to build your own. And you basically have to kind of group these genes based on known biological relationships, regardless of if they're in the same pathway or not. All right, let's get out of this one. Eventually, we're going to come back to this because I think these genes are key. It's just that they're missing a lot of key information. And I'm going to show you how to build your own pathways using this list as our core. And it's really cool. All right, let's click on day and cell type, 2A ANOVA.